So, as I was talking about the privileged stem cells. So, when I say the word privileged stem cells, then this property is uniquely harbored by mesenchymal stem cells. Mesenchymal stem cells are the other component of the bone marrow. So, in bone marrow, we are having the two types of stem cells. One is the hematopoietic stem cells. Okay, so hematopoietic stem cells are the stem cells which give rise to all the differentiated cells of the bone marrows like the WBCs, the RBCs, etc. Then another component is the bone marrow has is these mesenchymal stem cells. They can be easily found there, and also the other areas where the stromal components are there. So mesenchymal stem cells are the forms the the component of the stroma. Now, these mesenchymal stem cells, as I said, they are the privileged stem cells. So, when I say the word privilege, what do I mean? I mean they have this property that they can evade immune response. Okay, so when I say that they can evade immune response, this is a very important feature which I need to take into consideration when I am dealing with the patients where I have to either go for graft, where I have this fear of graft rejection, or in the patients of immunocompromised immune status. Okay, so and in the patients where they are having the severe immune deficiency, or they are having the pancytopenias, etc. So they have this inherent potential that we don't know how their immune system will react. So in these conditions, the property of evading the immune response and producing a therapeutic effect is essentially required. And when I am using these mesenchymal stem cells for the regeneration of any cellular component, then this property is fully served by these stem cells. So once I am harboring these mesenchymal stem cells and I'm then culturing them and producing through the genes, I'm reprogramming them to produce a particular type of cell, then they have this potential that they will not result into graft rejection or the immune rejection or any type of hypersensitivity response, which may lead to the deteriorating condition of the patient. Now, what these mesenchymal stem cells can differentiate into? Mesenchymal stem cells can differentiate into various types of stromal cells. They can differentiate into various type of stromal cells. So when I say they can differentiate into various types of cells, it includes that they can differentiate into bone or osteocytes. They can differentiate into cartilage or chondrocytes. They can differentiate into fat or adipocytes. Okay, and they can differentiate into muscle also, or myocytes. So you see that they can differentiate either into bone or into cartilage or into fat or into muscle. So this is the property holder mesenchymal stem cell. That is why they are called as multipotent cells. Multipotent stem cells, because they have this potential to differentiate into multiple type of cells of the different regions. Like they can differentiate into bone, cartilage, fat, or muscle. So that is why they are called as multipotent stem cells. So let's recall it. I told you two advantages of these mesenchymal stem cells. Firstly, they are immune. They have this immune property of evading the immune response. So they're immune privileged cells. Okay. Then, secondly, they are the multipotent stem cells. So they can differentiate into either into bone or into cartilage or into fat or into muscle. Fine. So this was all about the mesenchymal stem cells. Next, uh, we'll be talking about how these stem cells actually divide. So when I talk about the division of stem cells, so there are usually two types of division a stem cell follow. Either it could be a asymmetric division or it could be a symmetric division. Okay, so asymmetric means you have the stem cell, it will divide to give two daughter cells. Okay, one of the daughter cells goes and gets developed into a mature cell. Then it is the end of this life. It's terminated. Fine. Now this daughter cell does not go and differentiate into mature cell. Still, it has this capacity of self-renew. So it has this property of stemness. Continues to hold this property of stemness. Now in future, this daughter cell will further divide and give to two daughter cells, out of which one will have this potential of stemness and another will have this potential, can go and lead to form a mature cell. So this type of division we'll call as asymmetric division, where one daughter cells grows into a mature cell and other daughter cells continues to harbor this property of stemness. Symmetric division is when Symmetric division is when the mature stem cell divides, okay, and gives two daughter cells, and both these daughter cells have this property of stemness. So they don't, you know, get terminate into mature daughter cells. No, they still have this property of stemness or the stem cell potential, the property of self-renew. So this is most commonly seen in during embryogenesis. This type of division is mostly followed by the stem cells. So, during embryogenesis, the stem cells follow this particular type of division of symmetric division. Clear? So there are two different types of divisions a stem cell goes. One is asymmetric division and another is symmetric division. Asymmetric division is when the one of the daughter cells grows and terminates into a mature cell. Other has this property of self-renew. Symmetric division is when both the daughter cells have retains the property of stemness, and this is most commonly seen during embryogenesis. Okay, so this was all about the division of the stem cell. Now, another topic I would like to take now is the field of, as I was telling you, talk to you about the field of regenerative medicine. So, as the name suggests, regenerative. So, we are living in an era where uh, the molecular medicine has become very important part in the when we are dealing with the treatment of the patients, right? And also we have to deal with a lot of limitations when it comes to the transplantation, when it comes to the for example, if a patient has to go for a bone marrow transplantation or a patient has to go for the liver transplantation, then there are a lot of limitations. First limitations we have during this is the matching of the donors. You have to get a specific HLA matched donor. So matching of the donors becomes another limitation. So I just to say something called HLA. HLA is the human leukocyte antigen. So it is the gene or it is the antigen which is present on most of the nucleated cells and is responsible for the identification of the it's kind of a barcode with the help of which the body identify between the self and non-self. So if the body identifies something as non-self, it will result into the rejection. Okay, because the immune system will get activated and it will result in the rejection. So this becomes again the one of the that is why we always go for a matching of the donors before planning for any transplantation. Then second thing is the availability of these donors. Third thing is the environment in which this transplantation could take place. So it is not just ki I'll take the patient, I'll harbor the particular tissue, and I'll just transplant it. No, it requires proper monitoring. So a proper monitoring is necessary when we are talking about the transplantation. So there are just a few of them I've labeled, but there are similar kinds of other so many limitations, and also the immune status of the patient, whether the patient will be able to handle this transplant or not. So there's certain limitations when they're dealing with the transplantation. So when I say the regenerative medicine, what the concept this medicine has, we just talked about that the embryonic stem cells have this potential to self-renew, and they are totipotent. So they can differentiate into variety of cells, like they can differentiate into the cells of the endoderm, ectoderm, or mesoderm. Fine. But the major limitation is that the embryonic stem cells has this HLA gene. So this will result in the activation of immune system, and it can result into the most important thing, the dreadful complication of rejection. But I need a type of stem cell which can protect me from this type of response. So which type of stem cell we just read is the privileged stem cell. So we just talked about privileged stem cell as the mesenchymal stem cell. Okay. So mesenchymal stem cells, we just read about that mesenchymal stem cells, these are the privileged stem cells. So mesenchymal stem cells have this potential that they will not result into the activation of immune response. So they will have this potential of evading the immune rejection. So regenerative medicine is actually what they are trying. They are using this potential of this mesenchymal stem cell to differentiate, to harbor them, and to differentiate into the various type of stem cells, to mature cells, sorry. But I already told you here that the mesenchymal stem cells can differentiate into different types of stromal cells: bone, cartilage, fat, or muscle. Okay. Now this potential of mesenchymal stem cells to have the power as similar as that of the embryonic stem cell, like the totipotent stem cells, is a limitation. So now what the current medicine is practicing or planning to do, the researchers are going on that what we do, we harbor something called the native cell, the patient's own. So what we are planning is that the we are planning to harbor the patient's own stromal cells, or the uh, mature cell, and we are combining it under the laboratory preparation. We are combining them with the genes. These are the genes which have the property of stemness. So mature cell, for example, the fibroblast, when under the laboratory conditions have been grown with or cultured with the genes of the stemness, they will result in the formation of the uh, pluripotent stem cells. Now, these pluripotent stem cells will have the potential to differentiate into ectoderm, mesoderm, or endoderm. So what do we do? What do we call to this? This whole phenomena, we call it as induced pluripotent stem cells, IPS. So with if so this is under still under trial, but if we are able to do this, like if we take the mature 5 cells from the patient specific 5 cells and then they clone it with the genes of the stemness, we will able to have the cells which have this pluripotent potential, and then we can give will be successful in generating the various types of cells of the ectoderm, endoderm, and mesoderm. So what advantage it can have? For example, it can be advantages in the patients where, for example, if a diabetic patient is there, and we can generate with the help of this the insulin secreting beta cells. Fine. Another example is when we have in case of in patients with the uh, myocardial infarction, where the myocardium is damaged, then we can use these stem cells for the regeneration, which is, these myocardium is have very low intrinsic regenerative capacity. So that is why this, if this becomes successful, it will be very fruitful. Also in case of stroke patient, so in case of stroke patient, this can also be used for the proper healing, and because of the generation of the neurons again, if this potential can be extracted. So one thing you should always keep in mind, that this the myocardium and the neurons, they have very low regenerative capacity. So when I say they have very low intrinsic regenerative capacity, I mean they have a very limited healing potential. And what is regenerative medicine will do? Regenerative medicine will help in increasing their healing potential. Fine. So this is how this can be useful. So till now we have talked about the summary. I'll just quote the summary. We started our topic with the stem cells. We talked about the types of stem cells. We talked about the uh, how they are different from each other. Then we talked about their division. Then we talked about their role. Then we talked about the regenerative medicine, and then we talked about the induced pluripotent stem cells. Right? So this was today's class in which we talked about all these cells. Another important thing before summarizing my class, I would like to tell you the various location of these stem cells. So for example, they are talking about the adult stem cells. Adult stem cells, they are, if they are present in skin, they are basically located near the hair follicle bulge. And if they are present in intestine, they are present between the Paneth cells at the base. And for the liver stem cells, are also called as oval cells. They are present near the canals of Herring and the bile duct. Okay, etc. So there are many various locations of these stem cells. You can go and check on your textbook, and you can have the detailed idea over there. So this was all about a basic concept of the stem cells. I hope you like this class, and I'll be bringing the new interesting topics in future. And for any queries, any doubt, you can comment on my YouTube channel, Dr. Patho. You can tune in for the quick updates on Dr. Patho Shorts, and you can definitely reach me on my Instagram handle at Pathpal. See you guys soon. Take care. Bye bye.